So indeed, thank you, Rafa. We have two very nice speakers today. Starting with the first one, Dr. Joanna Banasiak. So Dr. Joanna Banasiak is an assistant professor in the Department of Plant Molecular Physiology at the Institute of Bioorganic Chemistry at the Polish Academy of Sciences. It's also in the Polish Academy of Sciences that she performed her PhD in the group of Michael Jasinski. Her research focuses on ATP-Binding Cassette transporters, which is an ancient and widely distributed family of membrane transporters that utilize ATP to translocate diverse substrates across biological membranes. And she's particularly interested in the ABCG transporters in legume plants and the roles that they fulfill in plant-microbe interactions, which she's going to tell us all about right now. So, Joanna, yes, the floor is yours.
Thank you very much for invitation and this introduction. So hello everyone, thank you for joining this webinar. My name is Joanna Banasiak. As was said, I am from Institute of Bioorganic Chemistry, Polish Academy of Sciences in Poznań. Poznań is one of the oldest Polish cities located in the western part of our country, well known in Poland at least, from its rich academic and cultural life, and our institute is localized nicely localized in city center. So the Institute of Bioorganic Chemistry conducts interdisciplinary research at the intersection of biology, chemistry, and informatics. Originally conducts its works focus on nucleic acid. Today covers much broad topics, including neurodegenerative and cancer-related diseases, but also plant and genomic and plant and human genomics, structural biology, bioinformatics, and of course, plant physiology. So today I will tell you something more about research, scientific interest and research of Department of Plant Molecular Physiology headed by Michał Jasinski, and our group is informally called Medicago crew, and we are mostly interested in ABC transporters belonging to G2 family in legumes. So I would like to today highlighting the role in plant microbe interactions during my presentation, present you our expertise concerning the functional characterization of this group of proteins. But first, let me give you a brief introduction to ABC. Protein family. Most members of this family act as active pumps that use ATP hydrolysis as a source of energy to transport substrate across membranes. They also share common structural features such as the presence of two types of domains, transmembrane domain TMD and nucleotide binding domain NBD. Two TMDs are involved in recognition and passage of the substrates through the membranes, while two NBDs are responsible for the binding and hydrolysis of ATP, powered the transport cycle. Based on the number of domains within single polypeptide, the arrangement, but also phylogenetic relationship, ABC protein family has been further divided into 9 subfamilies named alphabetically from A to I. Interestingly, ABC proteins are evolutionary ancient, very common, present in all living organisms. However, they are particularly abundant and diverse in plants compared to other taxa, and this may be due to remarkable adaptive plasticity of plants caused by the excessive life size, so the inability to relocate when exposed to unfavorable condition, but also huge chemodiversity resulting from development of secondary metabolism, the ability for post-embryonic development largely controlled by, of course, plant hormones, which also have to be distributed within plants, and finally, settlement of plants, which was preceded by the focal expansion of ABC proteins, which can be illustrated in the scheme presenting the distribution of ABC protein, whole families, and belonging to the particular subfamilies across different plant lineages, spanning whole archaeoplastida, including glaucophytes, red algae, and whole green plants. As I mentioned, this almost doubling the number of ABCs occurred before the emergence of land blends, transition between hierophytes and bryophytes. And this involved mostly the largest subfamilies, B, C, and of particular interest to us, G subfamily, whose members indeed are involved in many processes that support unique terrestrial plant lifestyle, such as formation of functional cuticule, but also establishment of arbuscular mycorrhiza, defense response against new pathogens, and of course UV light by secretion, secondary metabolites, and distributions of phytohormones. So as you can see, some kind of fundamental novelties which enable successful adaptation to land. So we already know about the important role of ABC transporters during terrestrializations. However, we decided to go a little bit further and ask the question about potential role of ABCG and the evolution biology of agronomically important legume plants, which characterize by quite unique biology, strongly related, of course, with plant microbe interactions. Legumes are able to establish symbiotic interactions with nitrogen-fixing bacteria, form more widespread and all their arbuscular mycorrhiza, and legumes are also particularly rich in secondary metabolites belonging to flavonoids class, with characteristic to them isoflavonoids and specific 5-deoxyflavonoids and 5-deoxyisoflavonoids, which of course play important role in plant microbe interactions. To study the role of ABCG transporters in legumes, we use model legume plant, Medicago truncatula. We have identified and functionally characterized several ABCGs, and today I will talk about 4 of them and demonstrated their important role in legume biology, but of course many more await for characterization in this context. Notably, in phylogenetic tree of ABCG transporters, here from 3 angiosperm plant families, Brassicaceae, Fabaceae, and Solanaceae, all identified and characterized transporters located within highly expanded clades called G40, with only one representative from Arabidopsis called ATP-Binding Cassette. This clade can be considered as a plant microbe interaction associated, as many transporters from this clade have been described as being involved in plant microbe interactions, both beneficial and pathogenic. In this clades, we can observe also expansion of ABCG transporters from Fabaceae, very often being a result of gene tandem duplication, what appears to have facilitated a functional specialization, what we postulated. So, and the good and first example of this can be G46, which plays an important role in the biosynthesis of legume-specific 5-deoxyisoflavonoids, presented here. We show that G46 is involved in plant basal defense response against fungal pathogen by influencing the biosynthesis of the main Medicago phytoalexin, medicapine, with antimicrobial and antifungal properties, which derive from phenylpropanoid pathway, more precisely from this legume-specific 5-deoxyisoflavonoid pathway. We have shown that expression of G46 is strongly upregulated after treatment with fungal elicitor, some kind of PAMPs or pathogen-associated molecular patterns, which also induce de novo biosynthesis of medicarpine. And in contrast to a methyljasmonate, a defense hormone, which is also involved in medicarpine biosynthesis, however, by remobilizing the vacuolar storage conjugated preformed precursors, and which does not affect G46 expression. And in consistence with this, silencing of G46 following elicitor treatment, dark green, decreases both accumulation and exudation of free medicarpine precursors, so-called aglycans, including the very early, so like isoliquiritiganin and liquiritiganin, and does not affect the accumulations of these vacuolar storage conjugated forms. So this results suggest the involvement of G46 in the early stage of biosynthesis of medicarpine, and indeed transport experiments conducted in BY2 cell overexpressing G46 performed by Wanda Biała-Leonhardt, our expert concerning transport experiments, LCMS analysis, especially phenylpropanoids. So transport experiments revealed that ABCG40 exports two compounds, liquiritiganin and 4-coumarate. Compounds we are located in this pathway in very interesting place, some kind of branching point. Both of them can be substrate not only for the biosynthesis of medicarpine, but also lignin, coumarins, and in case of liquiritiganin, also the 5-deoxyflavonoids, sorry. So we propose that the role of G46 can be, in response to fungal infections, can be redirecting these early precursors into the branch leading to the medicarpine biosynthesis. We are also interested in identifying some molecular determinants behind this substrate specificity or selectivity. So we combine different approaches, the example given by chemical studies, mutagenesis, structure prediction using AlphaFold2, etc. And we have identified transient access path, but the substrate to the central cavity, but also key amino acid which influence this substrate specificity and selectivity. Now I would like to back to this phylogenetic tree, since in this phylogenetic tree we could also observe a plant family specific expansion of ABCGs, which actually can be linked with the production and translocation of particular group of secondary metabolites, so flavonoids in Fabaceae, terpenoids in Solanaceae, and glucosinolase in Brassicaceae. So we postulate that in case, at least in an angiosperm, the expansion of ABCG transporters can be a result of adaptation to a specific chemical background. In this phylogenetic tree, we could also distinguish expanded and legume-specific subclades, and two expressions of two ABCGs from this subclade from Medicago is root specific, and their expression is highly induced after inoculation with symbiotic bacteria and Cifermeliloti, but also after cytokinin treatments. And interestingly, cytokinin for a long time were considered as a mobile signal orchestrating the events between epidermis, where infection occurs, and cortical cell layer, inner cortical cell layer, where nodule primordia are formed. However, the transporter responsible for the translocation of cytokinin at that time was unknown. We have shown that expression of G56 following inoculation with ensifermeliloti is not factor dependent, restricted to the infection zone, and expands from the stele to the epidermis and then also to the cortex. What is more, is strongly upregulated after exogenous applications of bioactive cytokinins. Transport experiment, this time conducted in collaboration with Markus Geisler from Freiburg University, performed using protoplast isolated from agro-infiltrated Nicotiana bentamiana leaves, revealed that G56 indeed is exporter of bioactive cytokinins. So the mutant, G56 mutant, but also RNAi lines, in the latter, the silencing construct was driven by tissue-specific promoters, so epidermal and cortical, exhibit decreased nodulation. The mutant also produced fever infection threads, suggesting the accumulation of cytokinin in epidermis. Moreover, we have also observed that cortical cytokinin response following ensifermeliloti treatment is abolished in the mutant, as indicated by the lack of induction of cytokinin response gene error 4. And based on this, we proposed a model in which G56 is responsible for the translocation of cytokinins, bioactive cytokinins, from epidermis to the cortex, and therefore preventing the negative effect of cytokinin on infection events, and in cortex promote cortical cytokinin response, and of course nodule organogenesis. Okay, so the functional characterizations of the closest homologue of G56, called G40, which also act as a cytokinin transporter, strongly suggest role of this transporter in translocation of xylem-derived cytokinins toward pericycle. This transporter negatively regulates both initiations of lateral roots and nodules. So it is surprising initially for us, opposing role of cytokinin transporters in nodule formation. We think it depends on the source of cytokinin, so epidermis or xylem, the direction of the translocation, and finally the site and timing of cytokinin perception, so inner cortical cell layer where cytokinin promotes cell division, or pericycle where cytokinin inhibits cell division and initiations of cell division. And last but not least, we are also interested in arbuscular mycorrhiza, as we try to identify the closest homologue of previously described PDR1 from Petunia, described in Enrico Martinoia laboratory. PDR1 is also ABCG transporters located within this mentioned expanded clade, is responsible for the secretion into the rhizosphere signaling molecules, trigoractons, that promotes AM formation by stimulating AM spore germinations and hyphal growth and branching. PAPDR1 is very specifically localized in hypodermal passage cells, so unsuberized cell located within exodermis, so suberized cell layer that forms apoplastic diffusion parietal. And the main motivation of our research in the legume context was observation that, unlike majority of angiosperm, many legumes, including Medicago, do not form these exoderms. So we decided to explore how the differences in root anatomical features can influence the earliest strigolacton-dependent step of AM formation. So first, we confirmed the functionality of PAPDR1 as a strigolacton transporter in Medicago. We have shown that Medicago expressing PDR1 exhibit higher rate of colonizations, exhibited also some strigolacton-dependent shoot phenotypes such as the deeper leaf margin serration, a shorter petiole, longer internodes. Then we combine the phylogenetic analysis and expressions profiling and selected as our most promising candidate MTG59, which is the closest homolog of PAPDR1 and whose expression is strongly upregulated under phosphate deficient conditions, which promotes strigolacton secretion, and after exogenous applications of synthetic strigolacton GR24. Expression of G59 is root specific, is predominantly expressed in outer cortical cell layer and the corresponding protein localized plasma membrane. And phenotypic analysis of G59 mutants revealed that the loss of function of this gene negatively influenced AM formation, especially at the early stage of interaction. But to confirm that the reduced colonization is indeed due to decreased strigolacton secretion, we used germination bioassay using seeds of parasitic wheat, Felipanca ramosa, which germination is completely dependent on the presence of strigolactons. So we have shown that the root exudates from the mutant exhibited lower germination inducing activity compared to the wild type. Yes, however, this concern only seeds being in some distance of the root, but not attached to them, suggesting that in case of Medicago, strigolacton can also passively diffuse. And based on this, we proposed a model comparing exodermal plants and non-exodermal plants like a Medicago. And we propose that in Medicago, strigolacton during arbuscular mycorrhiza in response to phosphate deficient conditions can be passively diffused, as they do not encounter this apoplastic diffusion barrier. However, to achieve the full extent of colonization, the action and active transport of strigolacton mediated by G59 is required. So to sum up my presentation, I would say that our group specialized in functional characterizations of ABC transporters in respect to various aspects of plant, mostly legume biology, so defense response, symbiotic association, also seed biology, and the context of the translocation of secondary metabolites, plant hormones, and signalic molecules. And one thing more, in the near future, I plan to investigate also the role of ABCG transporters recruited to mediate this time nodule-specific functions. I have selected some promising candidates, mostly based on phylogeny and expressions profile, and I'm really open and interested in potential collaboration. Especially, I will need help, example given with measurements of nitrogenase activity, especially in planta, so more sophisticated microscopy analysis. And conversely, if you or your research has identified some ABC transporter, we would be more than happy to help you with their analysis. And so in this picture, you find whole person involved in presented results. Our leader of our group, our boss, Michał Jasiński, Karolina Jarzyniak, Tomasz Jamruszka, Konrad Pakuła, our former members, and also Wanda Biała-Leonhardt and Aleksandra Pawela. We have a new member, Sara Blichasz, Michał Flewski, Krishna Priya, Anil Khan, and Maciej Nowak. Here I present also our collaborators, Enrico Martinoia, Renzo Borghi, Markus Geisler, and Jan Przewozowski. So from my side, it's all. Thank you very much for your attention, and I am open for your questions.
Thank you, Joanna, for a very nice presentation and for all kinds of fascinating insights into the ABCG transporter family. So indeed, as I already mentioned in the chat, should you have any questions for Joanna, feel free to post them there or to activate your microphone and ask them directly. Perhaps in the meantime, I can already start with a question. Imagine you have an RNA sequencing data set and you see all kinds of nice ABCG transporters popping up. And you kind of want to get an idea of what they are doing. Are there any ways of already checking this in silico, so bioinformatically, or is this still very limiting and difficult?
So I will say that it's one of I like it very much to do this, to get some sequences of ABC transporters. First, I will check the phylogeny, so check where they are located in the phylogenetic tree. Check what doing the closest homolog, analyze check the expression profile in silico. Now we have a lot of data, and that I try to figure out and just what they can do, and for sure I will have a lot of hypotheses that could be tested later. So if you have some interesting ABC transporters, please, for sure I will try to help you.
That's very nice. I see that someone's also typing in the chat. So if you have any questions.
May I ask a following question? A follow-up on this question. So do you think that single cell data would help you to have an idea of the role of the different ABC? Or have you already checked on the available data?
So for sure, yes. Example given this, I don't know, this one of these transporter, G40, which is responsible for the cytokinin translocation. We postulated that is mainly expressed in some particular cells in pericycle. So a single data set will help us to confirm, for example, this, and it will be really great, yeah.
I was also curious about, I've never thought about it before, the fact that in Solanaceae, the production of stragolactone would be at specific passage shell on the exodermis. Do you think it could be related to attractiveness or formation of hypopodium at specific sites?
Yes,
you expect a higher concentration of stragolactone at some part of the root surface? Do you think it's
Solanaceae?
Yeah, because exodermis is in Solanaceae but not in legumes, as you mentioned. So do you think it can play on the where hypopodium forms on the root surface?
Yes. Can I back to slides? Yeah, because there is some hypothesis that you see this. So this PAPDR1 stragolactone transporters localize very specifically this unsuberized cell. Yes. So, and there is some hypothesis that stragolactons transported by this PAPDR1 form some gradient of stragolactons, yes, and guide the hyphae to this entry point, which is unsuberized. So in Medicago, it's hard to say, most probably because there's no exodermis, no apoplastic diffusions barriers. are the ion-thangic and entry in whole roots. Yeah, so we propose something like this. It's hard to say if it's true. Probably we will need to do more experiments, but yes, it's probably the presence or lack of exodermis can influence the way how the hyphae enter to the roots. So Solanacea is limited, we think.
Hi, Joanna. I have also a question. You mentioned that the ABCG 59 could be expressed under phosphate starvation, or also if you apply GR24. So they can work like importer and exporter of strigolactone?
Rather exporter.
Okay,
because,
so how they are activated when you...
Okay, so actually in general, very often ABC transporters are induced by the substrates. That's why we test synthetic strigolactone. We also tested, for example, in the case of G46, the compounds from the dependent propanate pathway and works very well were induced. So it's standard for us. Cytokinin as a substrate of these transporters also induce expression of cytokinin transporters. So the second questions about the export of import, it's a little bit more tricky because, as I mentioned, it's maybe not mention it. In case of strigolactone, it's very hard to perform transport experiments. That's why we use this bioassay. Yes. But I think based on this bioassay, we said that G59 is responsible for the secretion of strigolactone, so export of strigolactone, similar to its close homologue from petunia.
Okay, and maybe this is a hard question, but you show a very nice role of different ABCG transporter for nodulation and mycorrhiza. Do you know if they are also involved in other kind of mutualistic symbiosis, like trichodermal? I know it's a tricky question.
No, unfortunately, no. To be honest, I don't remember any works that's linked to these transporters with other, but probably there's too less study.
Yeah. Okay, thank you.
Okay, I also have one final question that was lurking on my mind during the discussion as well. So you mentioned that in this big symbiotic family kind of in your phylogenetic tree that you see our plant microbe interaction clade, there's one Arabidopsis transporter as well.
Yes.
Is it known what that one transports?
Yes, very well. Yes. So this one is really well described. It's an abscisic acid importer. That regulates the and it's important because it's importer. It's that's obvious, and it regulates. Oh my gosh! So involved in drought stress, but also most probably as exporter of camalexin. And is related with typhus response. So it's very often ABC transporters can have more than one substrate. Sometimes, as you can see, camalexin and abscisic acids a different structure. Yes, so, because it is postulated that ABC transporters have multiple sites of substrate binding. So that's why.
Okay, that's incredibly fascinating. So I think one thing is clear, if anyone has any ABCG transporters that they're interested in, go ahead and contact Joanna.